Good morning, students, and happy almost end of the school year. Congratulations, congratulations. My name is Jamika Marshall, and I am the programs manager for the Shadow the Scientist initiative, which is based out of the University of California at Santa Cruz. We are pleased to be in the position to connect the community, students, the public, with scientists as they're engaged in research or engaged in activities related to research. And not only that, but it's in real time where you can engage with them, where you can talk with them, ask questions, and see the science in action. Now, today, it is my pleasure to present to you two amazing young ladies from the Massachusetts Institute of Technology, Jessica Bonnar and Mikala Capage. They will be sharing with you today and tomorrow about the amazing work that they're doing. Jessica, Mikala, take it away.
Awesome. It's so nice to meet you all. I'm really excited to be here this morning. So I'm gonna jump into the presentation that we have today. And I assume that your teacher has mentioned this, but this session is gonna be split over 2 days. So today is gonna be this sort of like lecture component where we're gonna be walking through the protocol, that I've been doing in the lab. And then tomorrow we're going to be actually imaging and collecting some data together live. So the project that I am working on is that I am imaging Drosophila larval ovaries. And Drosophila melanogaster is just the official scientific name for the model organism that are fruit flies. And so I think that we, already introduced ourselves briefly, but I'm Mikala on one side of the computer.
I'm Jess. It's really nice I'm not able to see everyone again, but I don't know if you remember, but I was in your classroom in December, so really excited to present to everyone. Really wanted to encourage, as we're doing the presentation, like please ask questions throughout. Obviously, we can't see you directly asking questions, but Mikala is going to be largely presenting today, and basically my goal is to see whenever Mr. Woodbury is like gesticulating at the screen to indicate that a student in the class is raising their hand for a question, and we really do want to answer them, and just like follow along in ways that are interesting for you. Also, at the end of the session, thank you for filling out the Google form. I've compiled all those questions, and we'll actually have you vote on the top ones that you want us to answer. And so then we'll go through it that way. But really, really excited to have you all here and to have Mikala presenting her work.
Okay, so before we actually get too much into the science, I want to tell you all a little bit about myself. So my name is Mikala. I'm from Oregon, raised in Portland, Oregon, and then I went to school. I did my undergrad at the University of Oregon in Eugene. Go Ducks. And then after graduating from the University of Oregon, I moved to Cambridge, Massachusetts, and started in the PhD program in biology at MIT. And I joined Ruth Lehman's lab at the Whitehead Institute. And so this is actually the building where I am in right now. And in Ruth's lab, as a graduate student, I knew that I wanted to work on questions about developmental biology. And so the broadest sort of definition of developmental biology is understanding how we go from a single cell zygote to a multicellular organism, and the really like complex process that has to occur in order for that process of development to happen. And so in Ruth's lab, I use Drosophila melanogaster, aka the fruit fly, to answer my scientific questions. And a lot of the work that I do is imaging based, microscopy based. So I threw up a little picture of a microscope here because I use microscopes of all different kinds in every single day in the lab. So now we wanted to, so I wanted to sort of start off a little bit with some of the fundamental, this like really important fundamental concept that underpins a lot of developmental biology, and that is that stem cells can become many different types of cells. And so up here on the screen is a little diagram of that idea where you have a stem cell and it has the capacity to become a cell in your nervous system, like a nerve, or a cell in your muscles. And I am actually in my lab particularly interested in stem cells called egg stem cells. And egg stem cells are capable of dividing to produce Drosophila eggs. And these are really specialized types of cells. Like I said, they produce Drosophila eggs. Those eggs are laid by the female fly, and then they go on to create the next generation. And so in my project, I focus a lot on these egg stem cells, and that's what we're going to be talking about today. So as Jess said, please jump in if there are any questions. I'm really excited. I love talking about stem cells. I think they are the coolest cells in your body. And so if there are any questions about those, please let me know. All right, so to tell you more about egg stem cells in particular, I want to tell you where they are located in the adult fly. And so this is a little schematic to tell you that sort of zooms in like deeper and deeper into where the egg stem cells are located in the fly. They are located first in the ovary, which takes up the vast majority of the adult fruit fly's abdomen. And then within the ovary, at the very tips of each of the two ovaries that a female fruit fly has, there is a specialized structure, which is depicted here on the furthest right side of the screen. And then at the very, very tip of this specialized structure, in this diagram, they're marked in orange. Those are the egg stem cells. And so they are located at the very, very tip of a very, very small organ that's located inside of the fly. And the egg stem cells stay in the ovary throughout the entire adult fruit fly's life. And they're continually dividing to produce Drosophila eggs. I looked this up because I wanted, so I was reading a little bit about this in preparation, and the female fly can lay up to 100 eggs a day from, just from these specialized egg stem cells. And so fruit flies live for maybe about 2 weeks. And so that is an incredible amount of progeny that one fruit fly can produce over her life. So if you ever wondered why your fruit fly infestations in your kitchen can get so bad, this is part of why. So I can tell you that egg stem cells are located throughout the adult life of the fly in the ovary, but egg stem cells actually have to first develop in the larval fly ovary. And so this is actually an image that I took with a microscope, and this is an image of what the larval fly ovary looks like. It's just this like ball of cells. And what we're looking at here is actually the DNA inside every single cell, actually inside the nucleus of the cell. And so that's what's marked in gray is the DNA of every single cell. And so the goal of all of my experiments in the lab is to count the number of egg stem cells. But if I just show you this image of the DNA inside the cells, inside the larval ovary, you can't actually tell very much. Like, you don't know where the egg stem cells are, and you don't know how many of them are there. And so basically, you can't tell very much about the ovary by just looking at the DNA. And so in order to actually understand which cells are egg stem cells and where they are in order to successfully count them, I have to do a protocol called an immunofluorescence protocol. And the goal of an immunofluorescence protocol is to identify egg stem cells. So on the left is an image of what a Drosophila ovary looks like without doing the immunofluorescence protocol. There's kind of no information. And after doing the immunofluorescence protocol, all of a sudden I can identify where the egg stem cells are, which cells are egg stem cells. These are the ones that are marked in pink in this image. And now, after doing an immunofluorescence protocol, I can count the cells. So what I'm going to be walking through for the rest of the presentation is basically, oh, yes,
Yes, we have a question. Let's have-
fantastic.
So how does the immunofluorescence protocol distinctualize between the egg stem cells and the other cells for you to be able to like properly observe it? Like how does it recognize those and only highlight those? Or
Yeah, were you able to hear that question?
I can. I might have you repeat some questions at some point, but I was able to
Yeah.
hear that one.
Okay.
That is a fantastic question. And that is what I'm going to be talking about in just a slide or two. So what I'm going to do is go through a couple more slides of the presentation, and then I'm going to follow back up and ask if that question has been answered.
okay.
Because that is like the heart of the matter. Like it's a fantastic question, and I hope that I will address it in the next couple minutes. And if I don't, tell me.
Cool.
Okay.
What other questions do people have?
Any other questions? Cool. All right, I think we're good.
We're moving on from part A of the worksheet. That's part A, stem cells. So does anyone have any blanks in that worksheet that we can just like help address right now and make sure are filled out?
Any blanks, you guys?
Yeah, I have one.
Yeah, I got a blank. Which bullet point?
It's the third bullet point, stem cells blank in the.
Ooh, stem cells blank in the blank fly ovary.
Is it like
Perfect.
Yep, it is developed. Stem cells develop in the blank. Yep, the larval fly ovary. So egg cells develop in the larval fly ovary. Any other blanks that you guys have? All right, looks like we're good.
Fantastic. Okay, so yes, as Jess said, we're moving on to part B of the worksheet. And what I want to take you guys through today is basically kind of what a week in the life in the lab looks like for me as I'm trying to do this protocol. So essentially, I want to do an experiment. I want to get a result. And before I can do that, I have to spend about a week in the lab working on this protocol. And so we're going to be going through it together and sort of step by step. And so on day one of this protocol, the first step is that you have to dissect Drosophila larvae. And here are just some pictures of what Drosophila larvae look like in the lab. And I actually should have included this life cycle image a little bit earlier in the presentation, because I really want to take a moment to highlight for you that, the life cycle of Drosophila is a cycle. It's very, very circular. So you start out with your adult organisms. Like I said, the female fruit fly is capable of laying about, up to 100 eggs a day. These eggs are single cells that will be fertilized, turn into a single cell embryo, and then undergo this really complex process of development, first becoming a small larvae, a bigger larvae, and even bigger larvae. And then because these are bugs, they have to crawl up the sides of their, whatever environment they're in. They form pupae, and then they hatch out of those pupae with wings as adults. And this process can start over again. And so the egg stem cells, again, are located in the female. The egg stem cells make eggs. Those eggs turn into larvae, and that's what I am dissecting. And so you can see in this little vial, larvae live in vials of food. They actually crawl down inside the food, sort of live and eat in there at the same time. And they live in these little vials that are plugged with cotton. That's to keep them from escaping, but also let enough air in. And this is actually an image of Drosophila larvae under a microscope, because they're really too small to see, really like finely see just with your eyes. And so these are larvae under a microscope, and I've got some little dissection tweezers next to them. And so on the very first day of this immunofluorescence protocol, I dissect the larvae. And this is a picture of what their tissue looks like from inside the larvae after I've dissected it out. And so then I do all of the rest of the protocol inside these little tubes on the tissue that comes from the larvae. So that's day one, which is to get your tissue from the dissected larvae. The second day of an immunofluorescence protocol is to add a primary antibody.
And maybe before we go into day two, any questions about the larva dissection? It's like super interesting, life cycle of flies, like anything related to that.
I'm assuming obviously you'd have to use a microscope to go in there and like fully dissect it. Yeah, which
Yeah.
can be tough because, you know, like when I was in college, I was working with nematodes, soil nematodes, and I had to extract them from the beaker and everything. And I know if I had like coffee earlier that day or like 20 minutes before, my hands were kind of shaky, and that's just amplified when you're, you know, having to work with all of that under a microscope. It's just going all over the place. So
Yeah. You definitely, you have to have really steady hands because you can
oh,
definitely,
exactly.
yeah.
Surgical hands. Yep.
Yeah.
Yep. It looks like we do have a question, Emerson.
Like, like entirely just physical implements to like divide up the larvae, or is there something specific you're looking for from the larvae? And also, do you use any chemical agents to like break them down, or?
Yeah, fantastic question. So to recap, I think the question was, am I looking for something particular inside of the larvae? And then is it all like manual, or are there some chemicals that are being used as well? So the dissections are all the process of actually getting the tissue out of the larvae, that's all like mechanical or manual. I'm using very, very small tweezers to manipulate the tissue and the body of the larvae to sort of get at what I want. And then that's a fantastic question as well about, you know, what am I actually looking for inside of the larvae? And so I'm going to move to this image. Basically, inside of the larvae at this point, there are kind of like three main things. There's the brain, there's the gut, and there's an organ that only exists in certain types of insects that's called the fat body. And the developing larval ovaries actually sit in the fat body. They're like physically attached to this other organ during development. And so the larval ovaries are so small that I can't see them with this level of microscope. And so what I do, and actually what's inside the tubes, is just the fat body of the larvae. And I'm basically trusting and hoping that I sort of captured the ovary when grabbing the fat body. I toss out the brain, I toss out the gut, I toss out the kind of like casing that's like holding the larvae together. I get rid of that as well. And basically, I'm just interested in the fat body because within the fat body should lie the ovary. Fantastic question.
And I will say, it took you how many months to sort of learn this dissection process?
So
we're talking about this recently.
Oh my gosh. Yeah. So I think I joined the lab, and then I spent like 3 months just like trying and failing and trying and failing and trying and failing to get these larvae dissected. And then by maybe like the fall of my first year in the lab, I was like, I'm pretty good at this. I had a meeting with my boss, and I remember writing in the slides that I was like, I'm pretty proficient in these dissections. And then I kept doing them, and like sometimes they work and sometimes they wouldn't. And I've been doing them for about 2 years now. And now I'm proficient. I don't know what I was before. That was not true before, because now I'm really good. And so it's been this like very, very long process. And it's been a lot of like trial and error. And it's been a lot of like stepping away and taking a break and coming back to it. And that is a really important part of experimental science. That's a really important part of research. And so it's very easy for me to just say like, oh, you just grab the fat body and all of that stuff. But it took a lot of work. And so I'm excited to be able to say so confidently to you all today, that this is how you do it. But it takes a lot to even get to that point.
Any other questions?
Any other questions? All right, looks like we're good.
Okay.
All right.
So day 2 in the lab. So I just dissect the larvae, I go home, I go to sleep, I come back. I start the process of doing the primary antibody. And this is where I should start to be answering that initial question about, how does immunofluorescence find egg stem cells? So let's hope that we're sort of answering that question. And again, if I haven't answered it, let me know. So essentially, primary antibodies, immunofluorescence assays identify cells based on the proteins that they contain. And so I'm going to introduce to you a very important protein in egg stem cells, and this protein is called VASA. And so what we have diagrammed up on the screen here is that egg stem cells contain VASA. We've diagrammed that for you as these like little gray clouds, and non-stem cells do not contain VASA. And this is actually a really sort of like sharp dividing line between egg stem cells and non-egg stem cells. Basically, the fly never messes this up. Its egg stem cells will always have VASA, and its non-stem cells will not. And so essentially, there are underlying differences between the egg stem cells and the non-egg stem cells in that the stem cells contain VASA, but I can't tell which ones are which because the VASA protein is not currently visible to me. And so that's where using a primary antibody comes in. A primary antibody specialized for the VASA protein will only bind to the VASA protein. So the first thing that I do in the lab is I have my dissected tissue, I add the primary antibody, and this is what primary antibodies actually look like in the lab. They're just liquids that come in little tubes. And so this one labeled VASA is this specialized VASA primary antibody. I add a very, very, very small amount of the liquid of this primary antibody to my dissected tissue. And so to go back really quickly, I've added this primary antibody, and the primary antibody is only going to bind to VASA. And so now I've created a difference in the larval ovary. The egg stem cells have primary antibody. The non-egg stem cells do not have primary antibody. But if I image those samples just after adding the primary antibody, it would still look like this. Because even though there's primary antibody added, I can't actually see the primary antibody. So basically, I've fixed one problem. I've added primary antibody, so I can tell like some of the cells have primary antibody and some don't. But if I can't see the primary antibody, then that doesn't really do me any good. And so the next step is on the next day, I go to sleep, I come back, the next step on day 3 is to add a secondary antibody. And it's at the stage of the secondary antibody where things start to actually become visible and get really exciting. So I've talked a little bit about primary antibodies, and I want to stop and see if there are any questions about primary antibodies so far.
Do you guys have any questions?
Oh,
Yep.
Do they develop with a lab somewhere? Like, how do you get specifically the antibody for?
Yeah, that's a good question, Emerson, because I kind of had a similar question to that. So the question is like, how do you get the antibodies that bind to VASA?
Great question.
Fantastic question. So fantastic question. Basically, antibodies in the lab take advantage of an animal's natural immune system. So this is gonna sound like this is coming out of left field, but imagine when you get sick with a virus or a bacteria, and you start to run a fever, and basically, if anyone's ever told you like, oh, that's your immune system like fighting off that invading like virus, that your body is making antibodies against a virus or against some sort of like invading pathogen. And this is a really incredible ability that all organisms have to generate antibodies that recognize invading proteins. And so in the lab, we've actually taken advantage of that natural ability in an animal. If you instead of like infecting an animal with a virus or some sort of like, something that will make the animal sick, if you actually inject the animal with Vasa protein, that animal's immune system will freak out and it'll develop these specialized antibodies that will recognize the Vasa protein. And so basically, the animal's body is trying to fight off this foreign protein. And we are, as scientists, are taking advantage of those antibodies produced by the animal by injecting a particular target. And so these are done with, small animals like rabbits, like that kind of thing, a lot of mice often. And then you take the antibodies out of the blood, and that is what is in this, like in each one of these little tubes. So that is a fantastic question about how we get antibodies. It's very complicated. But I hope that like sort of starts to get at the answer. It's this totally like crazy thing that we know we know animals do, and we're actually just taking advantage of that natural process to do our science.
And I do just want to make a note. Scientific research, especially in biology, often uses animal research and animals to help create things. And I've worked with mice historically, and it's something that is a big sacrifice to make, but there are a lot of protocols surrounding, like, if you are causing harm to the animal or if the animal is going through pain, there's a lot of steps that you have to go through to ensure that you're creating the least amount of pain possible, that you're really respecting the life of the animal. And for me, and how I was trained whenever we sacrificed mice, like, we really thanked them for like the sacrifice that we were making so we could better understand how the world works. Animal research is a really core part of science, and I think something that is deeply important with that is to like ensure that we are treating the animals with the highest quality of care that we can.
Yeah, excellent.
Great question.
Cool. So essentially, then the other thing that I would also want to say is that antibodies, especially because they come from animal research as well, are like a really valuable reagent, like resource that we have in the lab. And so, they're made like typically once and then shared with researchers like all over the country and all over the world. And so this Vasa antibody was actually made by my lab almost 30 years ago. And we share it with researchers literally all over the world. They will like email and say, can I get some of that like Vasa antibody? And so it is this like very, very precious resource that we can like take advantage of. And so I use like the tiniest bit of it every single time I do an experiment, because once it's gone, it's gone. It can't be made again.
And sorry, I'm going to add one more thing, and then definitely we should get to the secondary antibody. But one of those tubes that Mikala's holding right there, actually, if you wouldn't.
Oh, yeah, sorry.
Primary. Right, Vasa protein your lab made, but then I'm assuming maybe your lab bought the 1B1 primary antibody.
Yeah.
So, right, we just described this process of generating antibodies from animals. Like, you have to care for the animal and respect their welfare. And then there's a lot of like very important sort of purifying protocols. So that tiny tube of 1B1 probably cost around $600, right?
So I'll actually say that actually these are publicly available resources. So that tube of 1B1 costs $50.
Okay, great.
Some antibodies just
I regularly buy antibodies for $600.
Totally correct. Yes. Yeah.
And so I just mentioned that because people were starting to ask, like, what is the cost of doing science? And maybe we show you this tube of liquid and you go, it's a tube of liquid. But when you actually, I'm so grateful that someone asked that question of how do you get the antibodies, because there's so much like manual labor that goes into generating these reagents and
expertise, too, like the like knowledge that people who have these antibodies make to like do it and do it well, like that is worth, its weight in gold, basically. So
So science is expensive is what we're getting at. And there's a reason that it's expensive.
Yeah, expensive monetarily and also time-wise too, because, as you said, it takes a lot of skill to, extract these things from these organisms and to get it all, make sure you have the right antibodies and things from there. So really remarkable stuff.
Yeah.
I'm so glad that someone asked that question.
Yeah, a fantastic question. Because it doesn't really make any sense. It's kind of magic.
Oh yeah, it's immunology is very complicated, as I've been trying to tell my students, so they'll know. They'll know.
Yeah, okay, good. Yeah, that's all I know about immunology. Like we've actually maxed out what I know about it with what I said. So
Likewise.
yeah, super complicated. Okay, so we have our primary antibodies. Basically all we've done is kick the can down the road because I still can't see the primary antibody. And so this is where secondary antibodies come in. And basically, secondary antibodies have a fluorescent molecule attached to them. And the secondary antibody will bind to the primary antibody and basically light up wherever the primary antibody is. And so the way that I do this is that, again, this is kind of the state on the left that my larval ovaries are in after adding the primary antibody. I add the secondary antibody, and now the egg stem cells are going to be pink. And I can detect these cells because they now contain a fluorescent dye. And so this is what real life secondary antibodies look like in the lab. Again, it's just little tubes of liquid. These are fun because now they're bright colors, which is exciting. But basically, like, these are what the secondary antibodies look like. Again, I add a very, very, very tiny volume of these secondary antibodies to the tube. And the secondary antibody has now illuminated the VASA protein in the egg stem cells. And so this is what this is what the ovary looks like after adding the secondary antibody, where now there is, if I zoom in here, this is just a zoomed in picture from the bigger picture, you can actually now tell the difference between a cell that is pink, which means that it has VASA protein, from a cell, a nearby cell next door to it, which isn't pink because it doesn't have VASA protein, because it isn't an egg stem cell. And so this is, to sort of recap what an immunofluorescence protocol is doing. It starts with a protein of interest that marks a specific cell type that we're interested in looking at. In this case, for the egg stem cells, this protein was called VASA. We used a primary antibody that is specifically targeted to VASA, and it binds specifically to that protein and distinguishes between cells that contain VASA and those that do not. And then in the final step, we added a secondary antibody, which is attached to the primary antibody and is also attached to a fluorescent dye. And now the secondary antibody is lighting up the cells that contain VASA, which are the cells of interest. And so that is sort of the basic workflow for a secondary antibody. And I want to pause here because we have now completed part B of the presentation and the worksheet. I hope that I've answered that initial fantastic question from the beginning. But I want to pause here and see if there are any other questions about the process of immunofluorescence, about these fluorescent dyes, anything.
So far, so good, guys. All right, looks like we're all good.
All right. So I've told you about one protein that we look at. I've told you about the VASA protein for egg stem cells. But in the images that I actually take, I'm interested in getting a little bit more information from these larval ovaries. And so I actually do an immunofluorescence protocol against 3 proteins at the same time. And so now I'm going to tell you a little bit about what those other proteins are, where they're located inside the cell. So we've already talked about VASA protein marked in pink. And again, VASA protein is located in the egg stem cells kind of throughout the cell in the cytoplasm. But I also want to introduce this other protein in red called HTS. HTS is located in a specialized compartment within the egg stem cells, but it is also located around the membrane of other cells in the ovary. And so this diagram here is going to show you the difference between egg stem cells with VASA in their cytoplasm, with HTS in their specialized compartment. And then for non-egg stem cells, they have HTS on their plasma membrane, so around the border of the cell. And then I also am interested in this protein called traffic jam, and traffic jam is located in the nucleus of the non-eggs of the non-stem cells. So I've now added in 2 other proteins to focus on, and one of those proteins is like kind of particularly interesting because it's located in a different place between the 2 cell types. And so really quickly, I want to show you what the final product from this triple immunofluorescence protocol looks like. You can see in magenta is the egg stem cells, and it's a little bit hard to see because it's kind of overlaid, but in some of the egg stem cells, you can see these like bright red dots. And then you also see red signal on the membrane, and then you see green signal on the nucleus of the non-stem cells. So this is what it looks like in real life. I just want to go back to this like diagram and answer any questions that sort of come up, because we talked about the immunofluorescence for one protein, and now in reality, I've been doing this secretly the whole time on 3 proteins all at once. So any questions about these other proteins, things like that?
You got a question, Emerson? Yeah, go ahead.
Wait, why is it called the traffic jam protein?
Great question.
Yeah, so this is secretly the best thing about fly research, which is that we have, as researchers who study Drosophila, we have a lot of creative freedom in how we name our proteins. And often they're named for phenomenon that occur when you get rid of that protein. So often proteins are discovered by getting rid of them and seeing what goes wrong. And so traffic jam is named because if you don't have this protein, the eggs kind of like build up in the female fly and it causes a traffic jam. And so it was discovered that this protein was really important for kind of keeping traffic flowing within the fly ovary. When you get rid of it, traffic gets backed up, and so they named this protein traffic jam.
I love that because I was telling my students like, oh yeah, like we got Sonic Hedgehog protein, we got Pikachurin. So like, if you're a scientist and you discover something and how it works, you can name it whatever you want. And so they're like, yeah, Woodbury. But I'm so glad that you reinforced that and validated it. Another question, yes.
In the photo of the actual cluster of cells, some of the non-stem cells, I can't see like the green traffic jam protein being highlighted. Is that just an error of like the quality of the photo, or do some of those non-stem cells not actually have the traffic jam protein in them?
Fantastic question. Yeah, good observation as well. I sort of I simplified things for you a little bit, and I said, oh, like you've got your egg stem cells and you've got your non-stem cells. But in reality, there are a ton of different cell populations that are all considered non-stem cells. And those populations have differences in how they express traffic jam. And so this image is correctly capturing reality in that some of the cells have traffic jam and some of them do not. And so that's a fantastic point, that there's actually a lot of sort of heterogeneity, a lot of differences within the non-stem cell population. Traffic jam identifies some of them, but not all of them. Yeah.
And that's in part why Mikala is overlaying multiple markers, right?
Mm-hmm.
So because of that heterogeneity.
Yeah.
Cool. Good question. Any other questions that we have? Looks like we're good.
All right. So at this point, you should be able to fill out the entire worksheet. And so I want to make sure that if there's any sort of missing pieces within the worksheet here, that we have a chance to finish that out. And also if I, from any part of the whole worksheet, if I missed anything from part B as well, please let me know.
Cool.
And
Yes,
hope
you got it all? Yep, got a lot of thumbs up, so I think we're good on that.
we're hoping
Awesome.
this worksheet can serve as a resource tomorrow. So as your people are looking at the real-time experiments, they can sort of map back to what we've talked about today.
Yeah,
Excellent.
cool. Okay, so finally, on the final day of this protocol, I am image, and so that is what we're going to be able to do tomorrow as a group. And so I'm really, really excited. I'm really excited to image these samples. So I can't wait for tomorrow. I'm really looking forward to it. Okay, so there were so many fantastic questions provided in the Google form. And so I'm now gonna let Jess take over. Is that, yeah?
Yeah,
So I'm gonna stop sharing my screen, and we're gonna now have a chance to answer some of the questions that you all have already asked, or
yeah, that you've submitted in the Google form, and now we can decide how to answer them. So, Mr. Woodbury, I didn't check with you about this ahead of time, but is it okay if students take out their phones to scan the QR code here and do some voting? Cool.
Yep, that's totally okay with me.
So if y'all could take out your phones, scan this QR code up here, or go to pulleverywhere.com, pullev.com/Jessica B245. And basically these are all of the questions. Also, holding your phone sideways will allow you to see the full answers as well. But these are the questions. We got so many questions, they're so good. Can't answer all of them, but you can start to rank the questions according to like the one that you're most interested in us answering. And while you're going ahead and doing that ranking, and just to check, are people able to like respond in the form and submit an answer?
Hi guys.
You're able to? Cool.
We're getting confirmation. Yep.
All right, then I think we'll actually just start with some of these top 3 questions here. I think it's really interesting to talk about how people got involved in research, as well as how do you recommend people start doing research? And then I really like this third one. If you choose to do research, do you only have to do that for the rest of your career? So we'll start with those 3. And Mikala, do you want to start off how did you figure out how to get involved in research?
yeah. I first heard about the idea of doing research in a lab in my high school biology class, my freshman year of high school. I had a really fantastic teacher who had some research experience, just like your teacher, just like Mr. Woodbury. And so she was kind of always mentioning that, the people who learned everything that we were learning in the class had to do it through research. And so that's kind of how I first learned about the concept. And then I was able to do a little research experience actually in high school. I applied to a program that paid me $1,000 for 1 summer of research, which I think I was like 14. So that was like really exciting. That was a lot of money. And so after that, I was able to spend the summer being in a lab in Portland, Oregon. And then when I went to undergrad, I knew that I really enjoyed doing research, and there was a lot of research going on in my college. And so I joined a lab there as well. And basically just always kind of enjoyed what I was doing. And so I just always wanted to do more of it. So that's kind of how it first started for me. It was actually like in high school in a class.
And for me, figuring out how to get involved in research specifically, I also loved my AP Bio course. I had a great AP Bio teacher at Golden West. Go Blazers. Once I think a lot of people will start to get involved in research when they get to undergrad or if they go to undergrad. And emailing professors, I think when I was there, I talked about this a little bit, but emailing professors whose research you're interested in is really sort of like how I got involved in research. And I had to send a lot of emails, a lot of cold emails, and eventually I had a professor respond. And if you end up going to undergrad, most undergrad institutions like have a division of research, or most departments have a division of research. So that is how I went. I talked to them and sort of asked about research opportunities. And I think asking is so important, and it really is how it happens for you. Jamika, you are also involved in astronomy research. Are you okay with me putting you on the spot and asking how you got involved in research and how you started as well?
Yes, absolutely, absolutely. So, a bit of a circuitous path for me. I am a non-traditional student, so when I went back after my first round of grad school as a teacher, getting my master's degree there in education and teaching, I knew I wanted to be a scientist, and I wanted to work towards that. And so as I started working on my third degree, I found that for me, I didn't have those research opportunities. And I knew that the upperclassmen had them, but not for me where I was. And so I did exactly what Jess was talking about. I just started talking to people, and eventually I was able to come across people who were open to teaching me and to allowing me to learn from them and to get my hands on some data. So I'm doing that currently. I'm still a student, and I'm doing asteroid research, and it's absolutely wonderful and so fulfilling. But I wouldn't have, I don't believe I would have those opportunities if I had not asked and let people know exactly what I needed and what I wanted and what I was willing to do for it.
I think absolutely. And also, I don't know if Jamaica mentioned this, but you were a classroom teacher for 10 plus years?
13 years.
13 years, there we go. And I think this actually maybe gets into the second question that I wanted to answer before we get to, do you think being a scientist will be harder? Short answer, yes. But in terms of the question that was asked about if you do science at some point, do you have to be a scientist for the rest of your career? And I'll pop that off. First off, I just want to emphasize, I think Jamaica's experience is like a great example of how you can come to start doing research later in life, even if it's not immediately what you do when you're an undergrad. And we are both in a PhD program, which is like a very traditional route to doing scientific research. I think I mentioned this when I visited, but PhD programs are paid. We both make about $48,000 a year, which to some people is a lot, to some people is a little. For us, it is enough to be happy with roommates in Boston.
And it's more than the 1,000 that I first got paid.
Yes.
So I'm happy with that.
So PhDs in STEM fields specifically are paid for.
Mhm.
Many of our peers who are doing their PhDs right now are not going to go on to continue to do scientific research. In fact, one of our best friends who sort of got us connected to this program, she wants to go work in venture capital and specifically work in sort of finding funding opportunities for new startup biotech companies, right? And so she's not going to be doing research after her PhD. We have 4 minutes left, perfect. I also know people who go into policy and like really, really care about things like getting more funding for research, right? If a tube of antibodies is $500, we need money in order to be able to do these things. And then I know people who go on and actually they finish research and they go on to become classroom teachers. I have a really good friend from my old lab who got her PhD and really, really cared about doing more teaching. And so then she went and was a classroom teacher and taught AP Bio. So doing your PhD is like not the end game. It is a source of training. And then there's so many things you can go on to do beyond that.
Yes.
Since we have 4 minutes left, maybe let's do rapid fire, a couple more questions. First question, do you think being a scientist will be harder in the following years due to federal funding being reduced? Mikala?
Yeah, it absolutely will be. And I would say too that that like we are scientists, not particularly because it pays us very much, but because we love what we do and we care a lot about advancing human health. And so, you know, when federal funding gets reduced for science, it's not that, you know, we were making a lot of money before and now we're going to make less money. It's that we're actually able to do less work to advance human health even further. And so that is what federal funding goes to. And so you know, there are very real impacts that will happen and have been happening because of a lot of uncertainty with funding right now.
And the one thing that I'll just say to add on, I'd like to get to one more question. I mentioned that grad programs are paid for in STEM. That is through federal grants. So we get our salary directly through federal grants, and our graduate program admitted half the number of graduate students this year. So we're actually, federal funding is directly impacting the ability for people like us to go and get trained. And so it's really, and again, we make 48,000, which is a lovely amount of money, and it's more than I ever could have dreamed of when I was 14. And it's also not like the best paid thing in the world. So Really,
Yeah.
really hope that changes. How does signaling develop stem cells into further differentiated cells? Mikala, I think let's wrap up on this one. And then I'm going to take screenshots of all of this. And as we continue tomorrow, we'll continue to answer these questions.
Yeah, so actually,
Replace it. Yeah.
yeah, I am actually going to not answer that question, because it's very complicated, it's very exciting. But I would prefer if we would spend our last 2 minutes filling out this quick survey. We will have lots of time to chat because my microscope images are very slow. So we're going to be watching the scope do its work, but we're also going to have a lot of time to talk more. And so lots of questions can be handled tomorrow as well. So with your remaining 2 minutes before the bell rings and you get rushed out, please fill out this survey.
And to just wrap up, like Mr. Woodbury, thank you so much for letting us come and speak to your class. Thank you everyone today for asking such great
Fantastic questions. Yeah.
Please continue to ask them tomorrow. Tomorrow's going to be a lot of time, hopefully, to ask questions.
Mhm.
Thank you, Jamika, for being the heart and soul of Shadow the Scientist. And we're excited to see you all tomorrow.
Yeah. So
Thanks for showing up
excited.
despite grad night.
Yeah. Thank you so much. I appreciate it. I think my students got a lot out of it, and I'm pretty sure we're all excited for tomorrow. Yes?
Woo!
Yes. A very
All
emphatic yes.
right.
Fantastic.
All
Great.
right.
See you tomorrow.
Thank you. Thank you. See you tomorrow, and I'll see you in like an hour or two for my period 3 class.
Right?
Awesome.
Bye.
Bye bye.
Bye-bye.